someone let me know. I assume you can see my slides. So you can find the primary data, and you can reach me at these websites if you're interested in any of the papers or have questions afterwards. I want to tell you about a way to look at cancers as a kind of disease of geometry. And the most basic question we can ask is, why is cancer such a unique feature of living things? I mean, we don't have this phenomenon in modern robotics, for example. Why is this? And the reason is because biology uniquely uses something that our engineering yet does not. Most of our artifacts are a kind of flat architecture where the individual pieces are passive, maybe computational, but they don't have their own agendas. And this is not true in biology. In biology, there is, of course, this tiered multi-scale organization, molecular networks, cells, tissues, and so on. But it's not just structural. It's the fact that every layer of biology solves problems in particular spaces. So we call this a multi-scale competency architecture. Every subunit in its own space is trying to achieve something, whether it be simple homeostasis or something more complex than that. So what I'd like to tell you in today's talk is summarize this way, that basically, I'm going to tell you that all cells, not just neurons, communicate as electric networks that process information. This should be, of course, familiar to you because of the work that you all do. I'm going to argue that cancer can be detected, induced, and normalized by a kind of computationally guided molecular manipulation of electrical signaling that specifically focuses not just on molecular mechanisms at the cell level, but actually on large-scale cell cooperation towards specific anatomical goals. And boiled down to two sentences, the whole message is basically that just like the brain, your body tissues form electrical networks that store memories and make decisions. These are decisions about anatomy. And we can target now the system for control of the kind of large-scale collective intelligence that swarms of cells in vivo execute, and this has many applications in regenerative medicine and also, of course, to cancer. I'm going to show you all kinds of interesting, weird-looking creatures like this five-legged frog. This is not Photoshop. These are real things that we use in our group to investigate some of the signaling. So one thing that's often discussed in this field is what is the right level at which to address cancer? You know, should we be looking at single cells, at DNA, or some kind of larger scale control structure? And in particular, for example, people often will ask, why do human bodies have reduced regenerative potential compared to some of the animals like salamanders and planaria that I'll show you momentarily? And oftentimes what people will say is, well, the reason is to avoid cancer risk. The idea is that if you had a large body that needed to be around for many decades, and you had access to these plastic, highly proliferative cells, such as stem cells and so on, that are needed for regeneration, that basically there would be too much risk of cancer. And so a perspective focused on specific pathways, let's say cell cycle control checkpoints, TGF-beta, all these kinds of things, predicts that cancer and regenerative capacity should go together. That is, if the body has access to very plastic cells, there should be a high cancer risk. These pathways that allow proliferation for healing and regeneration should also be dangerous. So that way of looking at it suggests that mammals can't keep regenerative ability without the oncogenic cost. But there's a kind of a flip side to this, and you can look at it a different way. And if you have a perspective focused on morphogenetic control as opposed to single cell states, then you might predict the exact opposite. You might say that actually regeneration and cancer are opposite ends of the spectrum, because if you had good control over what it is that your cells are building, then they should be making healthy tissues and not tumors. And in fact, this is what the data say. The animals that are highly regenerative, in fact, have very low incidences of cancer. And I want to introduce you to one particular creature. This is called a planarian. So this is a flatworm. They are our direct ancestors, or similar to our direct ancestors. They have bilateral symmetry, many organs, all the same neurotransmitters that you and I do. And they have this amazing property that if you cut them into pieces, every piece regenerates exactly what's needed, so you get a perfect little worm. In fact, the old tissue actually scales down so that the whole worm is properly proportioned. If you cut them in half, this side makes a tail, this side makes a head. these cells were direct neighbors. So you know that, and yet they have the completely opposite anatomical fates. So you know that this is not a purely local decision because they're in the same location. They have to instead coordinate with the rest of the tissue. Now, this animal has two other interesting features. First, it's immortal. So there is no such thing as an old planarian. They do not age. They live for extremely long periods of time, and they don't have cancer. Now, you can induce cancer, but if you do that and you induce tumors and then you injure the animal so that it's forced to regenerate, it basically resolves, and so it goes back to normal as it rebuilds a normal body. And salamanders are vertebrates that have been shown to do something similar. So you see that regenerative repair and resistance to cancer actually go together. They're part of this incredible morphogenesis control machinery. So what I'd like to do in the first part of the talk is to think about what this is telling us. What does this mean big picture for cancer? And for that, we have to go back to individual cells, and we have to think about what it is that cells are doing and what do they try to do. So this is a single cell. It's called a lacrimaria. It has no brain, no nervous system, and yet it's incredibly competent at all of its local single cell type of activity. So its morphology, its behavior, its physiology, gene expression, all of these things are handled within one cell. And of course, its main agendas, as all unicellular organisms are basically to reproduce and make as many copies of itself as it can, and also to explore its environment and go wherever life is good. And if that sounds like metastasis, that it should, because that's exactly what I think is going on here. So what we have then is this fundamental question, which is that we all start life as a collection of blastomeres, so individual embryonic kinds of cells, and then during development, they work together to build something incredibly complex. So this is a cross-section through a human torso. You can see all of the tissues and structures in the right place, the right size, everything's correct relative to each other. Where does this information come from? Now, people are often tempted to say it's in the DNA, but of course we can read genomes now, and we know that there's nothing directly about any of this in the genome. What the genome actually gives you is descriptions of the micro-level hardware that every cell gets to have, so the proteins. And so we really need to understand, how do these cells know what to make using that molecular hardware? How do they know what large-scale structures to make? How do they know when to stop? And as workers in regenerative medicine, we want to know how to cause them to do it again. So if a part of it is missing, let's say during injury, how do you get these cells to rebuild what they already built once? And this is very closely tied to what happens when this process breaks down, which I'm going to argue is central to understanding cancer. That is, regenerative medicine is basically a strategy for resolving cancer. Once we crack this problem of controlling what it is that individual cells participate in collectives to build, then we will all automatically have a solution to the cancer problem. And now one might think that regenerative medicine is progressing nicely. We have lots of progress in molecular biology, genetics, molecular medicine, but there are some really fundamental knowledge gaps. Here's a very simple example. Here's a baby axolotl, and baby axolotls have little tiny legs. This is a frog larva or a tadpole of Xenopus laevis. They do not have legs at this stage. One of the things that we've done in our lab is to make frogolotls. That is, you can make a chimeric embryo that's part axolotl, part frog, to try to understand how the morphology of the animal is controlled. And now I could ask a simple question. I could ask you to predict whether frogolotls will have legs. You have access to the axolotl genome. You have access to the frog genome. Why can't we tell whether these things are going to have legs in advance? And by the way, if they do have legs, will those legs be made of just axolotl tissue, or can they convince frog cells to participate in making these legs? There's lots of questions that we can't even begin to answer. There are no models in this field that would answer that question despite having perfect genetic information. Here's another kind of remarkable example with all kinds of implications for cancer. Planaria, unlike the rest of us, most animals have Weismann's barrier. So if your body encounters mutations in its lifetime, those mutations are not passed on to your offspring, right? Planaria are not like that. Some species of planaria, the way they reproduce is they tear themselves in half and then regenerate. And that means that any mutation that doesn't kill the stem cell proliferates into the next generation and is expanded throughout the body. So for hundreds of millions of years of somatic inheritance, these animals have acquired an incredibly messy genome. In fact, they are mixoploid. So every planarian can have cells with different numbers of chromosomes. That's similar. You know, if you saw something like this in the body, you would assume that that's a tumor. That level of genetic diversity and mutations and so on, that is just complete chaos. And yet they have 100% anatomical fidelity. If you cut a worm into pieces, 100% of the time you get the correct anatomy. How is that possible, and what is that telling us about how many things we still do not understand about the relationship between the genome and the body, that these animals can do that despite their really chaotic genome? So we understand, we're getting better at the molecular mechanisms, but we really don't understand the algorithms of biology at all. So this is a central question, that, you know, how are individual cell decisions harnessed towards large-scale anatomical outcomes? And if we had the answer to this, we could basically, if we could control the behavior of this collective, we would have answers to pretty much every issue in medicine, with the exception of cancer disease. So we view cancer as one facet of a much larger issue: the control of the dynamic control of geometry by a collective behavior of cells. Now, regeneration is an amazing process that's very important to understanding this puzzle. This is a salamander, a Mexican salamander named an axolotl. These guys regenerate their eyes, their limbs, their jaws, portions of the brain, the heart. They regenerate their tails, ovaries, and so on. And what happens is, if you amputate, let's say, a limb at a particular place, and you can do it anywhere along the axis, it will grow exactly what's needed, and it will stop. When does it stop? It stops when a correct salamander limb has been achieved. So this is very important. As we think about cancer, the cancer has a number of aspects. For example, the fact that it's not building the right thing, but also cancers have been called wounds that do not heal. Basically, a failed process of regeneration, where not only is it not doing the right thing, but it doesn't know when to stop. So this stop condition is very interesting and important. It means that the system has to somehow understand or to be able to detect the error between the current state and the correct state, which means it has to know what the correct state is, in order to cease the very rapid growth, remodeling, and proliferation of these kinds of processes. Now, this is not just some weird thing that salamanders do. In fact, mammals do have some regenerative ability. So the human liver is regenerative, and that's been known since the time of the ancient Greeks. I have no idea how they knew that. Deer every year regenerate large amounts of bone, vasculature, innervation, and skin, up to 1.5 centimeters of new growth per day. So remarkably rapid regeneration. And even human children can regenerate their fingertips, although that with age that capacity is reduced. So what we would like to do is we would like to really move biology from where it is now, which if we look at the journey that computer science took, in the 40s and 50s, this is how you would program a computer, right? To make it do something different at the system level, you would literally have to rewire the hardware. You would physically have to change the hardware. And this is where all of modern molecular medicine is, right? We're very excited about genome editing and protein engineering and rewiring pathways and gene regulatory networks and so on. But all of that is still at the level of the hardware. We haven't even begun to exploit the power of manipulating the decisions that the collectives make and the actual software that's in the system. And one way to, and so I want to show you some examples of this, but one way to think about this is as the collective intelligence of cells in the cybernetic swarm robotics sense of intelligence, meaning problem solving by groups of agents towards a common goal. And a simple example is this. This is a normal tadpole. Here are the eyes. Here's the nostrils, the brain, the gut. And these tadpoles have to rearrange their face to become a frog. So the jaws have to come out, the eyes have to move forward, everything has to move around. And you might think that that somehow what the genetics does is specify the right direction and amount for everything to move, because then you can go from being a normal tadpole to a normal frog. So we made what are called Picasso tadpoles, meaning everything's in the wrong place. The eyes on the back of the head, the jaws are off to the side, everything is wrong. And we found that these animals basically become pretty normal frogs because all of these craniofacial organs will move in novel configurations novel paths rather, sometimes going too far and having to double back. But they keep moving around relative to each other until they get to their correct location for a frog face. So what the system actually encodes is a kind of error minimization scheme that in some sense has a set point, meaning the different organs know where they should be relative to each other. And there's feedback here that's not only on size control, but also on large scale shape. And so in my group, what we've been doing is this kind of feed forward scheme is the typical kind of standard mainstream way of looking at pattern formation, basically as a feed forward emergent process where you've got some gene regulatory network activity. They make some proteins. These proteins interact with each other, and then in highly parallel fashion, you get an emergence of something complex. But what's really critical to all of this are the feedback loops, which are not very well studied at all, which mean that when a creature is deviated from the correct target morphology, and that could be with injury, that could be with teratogens, with mutations, all kinds of things, there are feedback loops that kick in, and these feedback loops, both at the level of physics and genetics, try to reduce the distance in anatomical morphospace from wherever you are now to the correct pattern, meaning the target morphology for that species. So again, it's a typical homeostatic circuit. It's a kind of error minimization thing, but the set point is actually not a scalar like pH or something like that. It's the set point is a morphological distribution. So we're talking about navigating anatomical morphospace. And so what we've been doing, and what I'm going to show you, is trying to understand how the system remembers what the correct pattern is. And I'm going to argue that bioelectric networks are a really key part of this whole process. But one of the crucial things here is that the whole system works towards a specific goal. That goal being a location in anatomical morphospace, which is the correct pattern for that species. Now, the scale of those goals can change during evolution and during the animal's lifetime. So during evolution, we went from very tiny single cell scale goals, such as metabolic goals and so on, for the cells to cooperate towards very large scale goals, meaning keep growing until you've constructed this kind of thing, and then you can stop, right? So that's our set point, as opposed to a metabolic set point or something like that. But of course, during the lifetime of individuals, that cooperation can break down. And what will happen is that when cells get isolated from the rest of the network that binds them towards a common homeostatic process, they roll back to their individual single cell behaviors. So this is human glioblastoma. These cells basically have reverted to their ancient lifestyle. They treat the rest of the body as just external environment, because that boundary between self and world has now shrunk. They're no more selfish. Sometimes people in a game theory of models of cancer will say that cancer cells are more selfish than somatic cells. I don't think that's true. I think they're equally selfish. It's just that the self is smaller, right? So here, everything is being done in service of very tiny goals that belong to individual cells, cellular cells, whereas here the self is huge, and they're all quite selfishly following the stress reduction scheme of forming this kind of large-scale structure. So, and you can see all this in great detail in various papers. But the idea is that if all of this is correct, what we might be able to do is reconnect these cells to the network that operates this patterning program. So the idea is not to necessarily kill these cells, but to actually potentiate their working together towards normal morphogenesis, which requires us to ask, okay, so what are the patterning signals that bind individual cells with individual cell level goals into a large scale entity that's going to undergo morphogenesis? And so this is where bioelectricity comes in. So I will do this rather quickly, because I'm sure many of you already know this, but I'm talking about bioelectrics for the whole talk, not because I think that bioelectricity does everything. Certainly, it's just one layer of this complex morphogenetic field that impinges on cells in all directions in vivo. So, of course, there are chemical gradients, extracellular matrix, tensions and stresses, and other kinds of modalities. But bioelectricity is an important and I think somewhat unique aspect of this morphogenetic field, and it's critical throughout your lifespan. And the first thing that I want to show, and this is a very kind of simple view at a single cell level. So in the plasma membrane, you've got these ion channels, and these ion channels are allowing charged molecules to move in and out, and as a result, they will acquire a voltage gradient across the membrane, which we call Vmem. So it's basically the transmembrane voltage potential. And at the single cell level, it's now known, and many details are here, it's now known that this kind of resting potential controls various cell level properties. Okay, and in particular, one thing that you can do is throw a bunch of different cell types on a scale of voltage, and you see something very interesting, that kind of terminally differentiated quiescent adult cells end up out here, so pretty hyperpolarized, whereas down here in this depolarized region live embryonic cells, stem cells, and cancer cells. You'll notice liver is kind of unusual. It's a mature organ, but it hangs out with this plastic crowd down here. So maybe that is interesting for liver regeneration. So there's clearly a an interesting relationship here. But one of the most important things is I often hesitate in showing this slide in talks, because people get very attached to this idea that this voltage is what determines cancer, and that's true at a single cell level, but it's actually much more interesting than that. The real power of bioelectricity is not in the single cell state; it's in the ability of these networks to process information. Now, how do electrical networks process information? Well, we can take most of our concepts from neuroscience. In neuroscience, they have this very nice understanding of hardware and software. That is, the hardware of the brain are these neurons that have ion channels. They set voltage states, and then they can communicate those voltage states to their neighbors in a flexible way. And the commitment of neuroscience is that these networks do all of the information processing that are required for cognition, so decision making, so plans, large-scale goals, the kinds of things that drive a coherent organism that's made up of cells, right? So we're all sort of piles of neurons and other cells. How is it that we have unified goal-directed activity? So neuroscience tells us that these electrical networks are the glue. They're the cognitive glue that binds individual cells into large-scale behavioral programs. And you can, using neuronal decoding, you can actually try to get voltage data from a brain. So this is this group did this lovely voltage imaging on the zebrafish brain, and you can try to decode what the information is. So remarkably, it turns out that all cells in your body do this, right? So this is not just for neurons, and we can have the same kind of research program where we can try to identify the electrical conversations that go through these networks and try to read them and understand what they mean. And this is a voltage-sensitive fluorescent dye that I'll talk about momentarily, showing you in non-neural tissue. In fact, long before neurons even appear, already these cells are taking advantage of computation via bioelectrical cues. So what we did was we developed a few techniques to try to track this in vivo. And so this is one example. This is all using voltage-sensitive fluorescent dyes. This is an early frog embryo. It's a time lapse of an early frog embryo putting its face together. And so you can see here, this is one frame from that video. You can see that long before the genes come on and the craniofacial organs are demarcated, you already have this ectoderm already knows where it's going to put all the different organs. Here's where the animal's right eye is going to go. Here's the mouth. Here are the placodes. Everything is already set up. And I'm showing you this pattern because it's the most obvious one. We have others that we found that are quite difficult to decode. But here, it's very straightforward. You can see exactly where the different organs are going to be. And we know this pattern is instructive because if you move any of these, not the cells, but the electrical state, if you change the electrical state, then the craniofacial pattern will follow, and I'll show you that momentarily. So these are native patterns that are required for normal development. In fact, human channelopathies, human patients with mutations in various ion channels have craniofacial brain, gut, limb disorders exactly like these model systems. So we know they're essential for normal development. This is a pathological pattern, and we'll talk more about that. What you can see by injecting a human oncogene, you can see that before even the tumor shows up, early imaging shows you that these cells are already bioelectrically disconnecting from their neighbors, right? You can tell they're about to convert. So we have these tools for detecting these bioelectrical signals. We have computational tools for understanding how it is that voltages arise from the different ion channels that are expressed. And the most important thing is now we have techniques for manipulating these electrical states in vivo. So we do not use any applied fields. There are no electrodes, there are no frequencies, there are no magnets, no electromagnetic radiation, nothing like that. This is all molecular physiology targeting just two things: the native machinery that cells use to communicate electrically. We can target the topology of the network, basically who talks to whom, by opening and closing gap junctions, so these electrical synapses, and we can do that with drugs, with mutations, and so on. Or we can go in and actually change the voltage of specific cells by targeting these ion channels. And again, we can do that with drugs. We can put in new channels. We can remove channels. We can open them, close them, and we can use optogenetics. So we can use light to trigger light-sensitive ion channels that will let us put down light masks and set whatever voltage pattern that we actually want. So what happens when you do this? When you start manipulating this information, you can do things like this, and these are just a few examples. So one thing you can do, I told you that craniofacial, that electric face pattern was instructive. Well, one thing you can do is you can look to see what the pattern is that kickstarts normal eye development, and you can say, well, what if I introduce that same pattern somewhere else? So this is an early frog embryo. We take some RNA encoding a particular potassium channel that we chose that was going to set these cells to the same pattern that normally kickstarts eye development. And we can introduce that, for example, into cells that are going to become gut. So here's the endoderm-derived gut. And sure enough, if you introduce that pattern, the local cells, using that pattern as a reference, they will build an eye. And these eyes can have all the same internal components as Normalise, so retina, lens, optic nerve, all that stuff. And there are two interesting things to note here. Number one is, note the modularity. We did not tell all of these cells how to make an eye. This is not about setting individual cell fates or differentiating stem cells or anything like that. In fact, we have no idea how to make an eye. It's a very complex organ. What we have found is a biological subroutine call, which happens to be an electrical state, that triggers the eye building module. So this is very convenient for regenerative medicine because it means we can kickstart self-limiting, very complex organ building cascades long before we actually know how to micromanage them. In fact, I don't think we want to micromanage them. The other interesting thing about this is that this is a cross section through a lens in the flank of an animal. The blue cells this is beta-gal lineage label. The blue cells are the ones that actually got the ion channel injection. These other cells are their neighbors that they recruited into making this nice lens, because these cells are not enough to make a lens. So there are two levels of instruction. There's us instructing these cells that a lens is what belongs here, and then those cells instruct their neighbors saying, here's how many cells we need to make a proper round lens. So you can see this amazing organizing principle that we can start to take advantage of. If you do that kind of thing, you can make ectopic otocysts. So here, this is the inner ear kind of organ in the frog. You can make ectopic hearts. Here they are. You can make ectopic forebrain, and then ask whether these animals are any smarter than controls. You can make ectopic legs, as you see here. And of course, you can make ectopic fins. I don't have time to get into this, but one of the things, the tadpoles aren't supposed to have fins, and we've shown that you can actually call up organs from other species. In other words, organs that are not within the normal target morphology of the animal, by pushing the electric circuits into attractor states that basically belong to other species. You can use it to explore the anatomical morphospace of the animal using the exact same hardware of the cell. So you can also use this to repair very specific birth defects. So this is a bioelectrical pre-pattern of the early brain. So this tells the animal how big and what the shape of the early brain should be, and it has this very characteristic kind of bell curve pattern. And what happens is that there are many teratogens that mess up that pattern. So basically, nicotine, alcohol, all kinds of mutations, many things that give brain damage, in fact, do it by turning this nice bell-shaped curve into a flat line, either hyperpolarized or depolarized, but you lose this very specific shape to the brain pre-pattern. And so what we did was we created a computational model, and this is work with our collaborator Alexis Pytak. We created this model that basically asked the following question: if we can understand what the bioelectric network is that is responsible for this wrong pattern, could we fix it? are there ion channels that we can open and close to force this network to go back to this nice bell curve shape. And it turns out that you can. If you interrogate the model for different channels that will allow you to do that, what it said to us is that there's one particular channel called HCN2, which has this very interesting voltage sensitive property where it serves as a kind of almost a contrast enhancer. So basically, when you have a flattening of this pattern, it will actually amplify parts of it to try to go back to the correct shape. And when you do that, something amazing happens, and you can do that in two ways, with either drugs that open native HCN2 channels, or you can simply put in new HCN2 channels into the animal. Here's what a normal brain looks like: forebrain, midbrain, and hindbrain. These animals have a mutation in a gene called Notch. So this is a very important neurogenesis gene, dominant mutation. You can see the forebrain is gone. The midbrain and hindbrain are basically a bubble. They have no behavior. They just lay there doing nothing. It's a very, very harsh brain defect. But on top of that, or in fact, on top of a variety of perturbations with nicotine and alcohol and so on, you can get back to a perfectly normal brain function, including in addition to structure and gene expression, such that they get their IQs back. These animals are indistinguishable from controls in learning in terms of learning rates and so on, even though they have that mutation and or they were exposed to these drugs, because the bioelectric intervention, meaning opening these specific HCN2 channels or introducing them, got the brain pre-pattern back to the right shape. Okay, and so this is a very specific repair of a complex defect by using a computational model that tells you which drugs to use to get the bioelectric pattern back to normal. The pattern not of individual cells, but the distribution across the early ectoderm. So now we're also using this, of course, for regenerative medicine, where organs are simply missing, not just malformed. So here's a frog. Frogs, unlike salamanders, normally do not regenerate their legs at this stage. So you amputate, 45 days later, there's nothing. But you can make a cocktail of bioelectrical kinds of drugs that basically will it basically will it basically will induce, it will trigger the whole regeneration cascade. So it turns on pro-regenerative genes such as MXX1, and then immediately by 45 days you can already see there are some toes, there's a toenail, and eventually there's a very nice leg that's formed that's both touch sensitive and motile. So you can see the animal can feel the touch and uses the leg, and eventually it becomes a very nice looking frog leg. So we are at the moment moving forward with that technology into mice, and trying to drive towards some sort of clinical application. I should do a disclosure here. David Kaplan, who makes these wearable bioreactors, he and I have a company that we've spun off called Morphoceuticals Inc. And the idea is that by these wearable bioreactors, we can introduce payload to the wound, which are basically mixtures of ion channel and other drugs that will kickstart a build a leg here kind of module, right? Again, the idea is to trigger a complex response, not try to micromanage it, but basically convince the cells that that's what they should be building. And all of this does work. This is not just something for frogs and worms. These kind of strategies, we've used them in human mesenchymal stem cells and cardiomyocytes to do these kinds of things. But of course, the most important thing is the context, the in vivo context. So what I've shown you so far is that, like the brain, the mechanism that binds cells towards large-scale purpose in morphospace are bioelectrical networks. And modifying that electrical communication gives you some really remarkable control over growth and form, which is very hard to achieve bottom up in terms of genomic editing and things like that. And so now I want to show you the data for applying this kind of technology to cancer in particular. And so there are basically 5 kind of main points that you might want to make if electrical signals were important for cancer. First of all, there should be physiology data that implicate bioelectric parameters, and there should also be data implicating ion channel and pump proteins in cancer. And then you want to be able to use this bioelectric signature as a tool for tumorigenesis, to observe nascent tumorigenesis. You want to be able to show that you can actually induce that kind of phenotype without genetic damage by modulating voltage gradients. And then most important of all, you want to be able to suppress or reverse the cancer phenotype with that strategy. This I've already shown you. We've already sort of looked at that. These data are actually quite old. I need to now at this point update them. But you can see here that that the field of ion channels in cancer is growing quite nicely. There are now over a dozen genes that are ion channel type genes that are known either oncogenes or tumor suppressors. And people do things like this. You can see that during progression. So this is just data from the GEO database. During progression, there are very clear changes in, for example, ion channel expression. But this is really important. Focusing on this aspect of it, I think, is holding the field back, because the key here isn't the gene. It's not one specific gene. It's not the gene product. It's a physiological state, the bioelectrical state. That's the key parameter, and in fact, not even a single cell state. And so let's take a look at what that looks like. So here's what I've already shown you, the this idea that bioelectric signals can be used to detect early stages of transformation, and you can kind of see how it compares to existing modalities. But one of the things we're working on is a kind of diagnostic technology with these voltage dyes, so that either at home or in the doctor's office or during surgery, you would be able to put the dye on some tissue, and you could see not only areas of tumor or pre-tumor, but you would actually be able to see where the margins are, right? So this is a kind of vision of a kind of augmented reality device where on top of whatever the surgeon is seeing, you can paint the bioelectric state that is inferred from the voltage dye data that you've sort of sprayed into the wound, right? So this is one of the things we're working on. Okay, so this is the detection part. Now the question is, can you actually induce this kind of phenotype without genetic damage? And you can. So this is a tadpole. These little black cells are melanocytes. So these are the normal pigment cells. And one thing you can do in animals that have no genetic damage, no exposure to carcinogens, no mutations, nothing like that, you can simply use an ion channel drug to modulate how the melanocytes talk to another cell population, which we call the instructor cells. If you do this, something amazing happens. These melanocytes go completely out of control. That you can see here the difference. They colonize the whole animal. In fact, if you take sections through those animals, you will see that the melanocytes, instead of a small number of little round cells here, this is the neural tube. These are the lateral tissues. Instead of that, what you see is this. You see these cells, they change shape. They become incredibly invasive. They're trying to spread throughout the body. They've already dropped into the lumen of the brain. Here they are in the actual neural tube, and here they are in blood vessels. This is metastatic melanoma. These animals these converted melanocytes are invading the blood vessels. They're doing all the things that melanoma would do in this model, but there is no genetic defect. There is no primary tumor. All of the melanocytes are doing this because they're confused about what they should be doing, because we've prevented them from talking electrically to their neighbors. You can do this with human melanocytes in culture, but there's something very important. This is not about the voltage of the cancer cell itself, because here, this is a cross section through one of those animals. These are the melanocytes, and you can see they're spread out. They're completely abnormal, and they're converted. The cells that we targeted are down here in blue. These are the cells that have the abnormal voltage potential, not the cancer cells themselves. So it's a long range effect that is all to do with how these cells receive instructional signals from their neighbors. Okay, so it's the voltage change in the cell's environment. You can think of it as microenvironment, except it's not really microenvironment. It works at a considerable distance, in fact, across the whole animal. That is a physiological switch towards metastatic behavior. And I don't have time to go into it, but we know how the switch works. It has to do with the movement of serotonin actually between these instructor cells and the melanocytes that take them through this pathway that turns on slug and SOX10 and all these things towards metastasis. And so all the genes that are downstream of this conversion are now known. So of course, inducing cancer is instructive for the mechanism, but what you really want to do is prevent or convert it, and we can do that. So if you inject any typical human oncogene, let's say a nasty p53 mutation or a KRAS or something like that, you will get tumors, and these oncogenes are labeled with a red fluorescent protein. So you can see here it's very strongly expressed right there. But if you co-inject an ion channel, in fact, you can place this, it doesn't even have to be in the same cells, you can place it in a different location. But if you trigger the appropriate bioelectrical communication and you force these cells to remain part of the electrical network that specifies morphogenesis, instead of having them go off on their own, which is what the oncogene does. The first thing the oncogene does is try to depolarize cells so that they disconnect from their neighbors. If you force them to remain connected, even though the oncogene is very strongly expressed here, in fact, it's all over the place, there's no tumor, because what guides actual cell behavior is the physiological state, not the genetic state. So that's very important. This divergence of genetic information from physiological information is key, because we want to be tracking the actual driver, right? We want to be targeting the actual driver. So if you do that, you can also do that with light. So with optogenetics, we've done this using light to turn on specific channels to ameliorate the oncogenic phenotype. And in fact, you can even, this is to really kind of drive home the point of how important the environment is. As a normal tadpole, this is a tumor sitting in the edge in the tissue of a tadpole, and you can see it's already starting to sort of reach out and metastasize. This is an eye. This is an ectopic eye forming in the head. The difference between making a tumor and making an eye is how much sodium a particular channel is putting in. So the amount of ions in the medium, okay, the amount of sodium that's going in, because of course it controls the resting potential, spells the difference between making a healthy organ such as an eye and making a tumor. So it's really critical to understand these bioelectric states as the drivers. So we've been moving all of this to human medicine. This is a most recent paper that just came out a couple of weeks ago. You can see this. We did this in vitro, both in rodent and human cells. And what we did was to choose and test a variety of human-approved ion channel drugs. These are things people already take for their ability to stop glioblastoma growth, in some cases even after the drug is removed. And one of the things that you see here is that what these drugs are doing is cranking up expression, among other things, cranking up expression of connexin 43, which is a gap junction protein. So that connectivity between the cells, that electrical network is starting to be built up. So the future, we think the one roadmap for this field looks something like this. There should be a platform for picking electroceutical blends. So basically, what will happen is that we already know from transcriptional profiling, we already know what channels are expressed in different cell types. We need to do a lot of physiomic profiling to understand what voltage state is required in any particular tissue. So what's the correct pattern? And if you know what the correct pattern is, and you know what channels exist as your control knobs, you can ask the simulator, and we've created this bioelectric tissue simulation engine. You can ask the simulator to say which of these channels do I need to open and close to get to the correct state. And once you know what those channels are, then you can pick either from off the shelf or discover new drugs that would open and close the channels exactly in that way as predicted by the model. And so I've already shown you an example of this. That was the brain repair. So that's exactly what we did in the brain repair. We asked which channel could be used to repair the bioelectric state, and it gave us a drug, and it in fact corrects brain patterning. So the idea is that all of the existing drugs that are target ion channels, and something like 20% of all drugs are ion channel drugs, form this really important class of electroceuticals, because lots of these are already human approved. People are already using these compounds, so the safety data exists, and we've begun creating this computational platform where you can get on and pick your tissue of interest, and it knows all the channels that are there, and then it will be integrated with this profiling and the Betsy bioelectric simulation engine to actually tell you, now that you know what these channels are, which of these do you want to target with your blend. So the summary of this last part are that cancers are not just genetic diseases. They're really a disorder of the scaling of cellular competencies in navigating anatomical space. And the bioelectrical properties can be used to detect, induce, and reprogram these kinds of behaviors without necessarily touching the genome, exactly as happens in the nervous system, because this electrical activity is in fact the computational glue that binds individual cells towards making healthy organs and away from metastasis and tumorigenesis. And we're now working on these pharmacological and optical strategies to exploit that property and to basically strengthen the ability of cells to work together. So kind of our future goals are to refine this physiological signature for better diagnostics, to move our VMEM control methods into mammalian systems, so mice first and hopefully then clinical, and to really crack this bioelectrical code, to really be able to induce normalization without trying to kill the cells, to exploit the kinds of ion channel drugs that are already in medical use and available to patients. So in the last couple of minutes, I want to show you an interesting thing that doesn't look like cancer, but it's actually, I think, quite related. If you're not yet convinced that your body cells have individual agendas, we can ask a simple question. To see if it's really true that your body cells would rather be doing something else if they were freed from the instructive interactions from their neighbors. So let's talk about this issue of what the default state of your body cells are. You might think, let's think about your skin cells. Now you might think that your skin cells have a predetermined kind of life. It's sort of boring. It's two-dimensional. They're the outer surface that protects you from pathogens and things like that. And you might think that that's all that the skin cells really can do or really want to do. And so we have a synthetic morphology project that addresses these kinds of questions, that talks about the role of the genome in setting those kinds of goals and the baseline behaviors. So here's what we did. We took some skin cells from an early frog embryo. So this top region here, these are just skin cells, and we asked the simple question: what would happen if we liberated them from the rest of the embryo? We don't add anything. There are no transgenes, no weird drugs, no nanomaterials, nothing. We don't add anything, but we do subtract something. We subtract the instructive influence from the rest of the body. And we ask, what do skin cells actually want to do if they're on their own? Now, we put them in a dish. They could do lots of things. They could do nothing. They could crawl away from each other. They could form a monolayer, like in two-dimensional tissue culture. They could die, many things they could do. Instead, what they do is, you can see them here, these dissociated cells are plated inside this little depression. They overnight, they come together here into this little ball, and they start to flash these calcium signals, which are very brain-like, although there's no nerves here, there's just skin. But they start to flash these calcium signals indicative of their computation, these network-level computations. And what happens then is they make what we call xenobots. Xenopus laevis is the name of the frog. These are biobots. So xenobots have a bunch of capacities. They can move on their own. The way they're moving through water is to use little cilia, little hairs that they have on their surface, which are normally used to redistribute mucus on the surface of the frog. You can see here they can go in circles. They can patrol back and forth in groups. They have all kinds of behaviors. They can go on these kind of long journeys. They can interact with each other. They can do nothing, and so on. Here you see one navigating a maze. There's no movement of the water here. This is perfectly still water. It can take the corner without bumping into the opposite wall, so it can tell there's a corner here. It turns that. Then at this point, for whatever reason, spontaneously it turns around and goes back where it came from. Right. So they have spontaneous behaviors that are not paced by us at all. They can regenerate. So if you cut them in half, they will bend through this. So you can see here. So basically, cut it in half, and then they fold through this hinge to try to regain their shape again. Again, they have these really interesting brain-like calcium activities, and then perhaps the most amazing thing is these. What we've done is we've said, well, these xenobots can't replicate the way that frogs normally replicate. We've made that impossible. But instead, if we sprinkle loose skin cells into their environment, what they basically do is von Neumann's dream, which is a machine that runs around, collects parts, and makes copies of itself. This is kinematic replication. Like us in making the first xenobots, what the xenobots do is they collect these cells into little piles. They polish the little piles into little spheres, and then the competency of the cells take over, and those little spheres become the next generation of xenobots. So each generation of xenobot produces the next generation. Once the little spheres are mature, they start running around and doing the exact same thing and collecting cells in the environment and making more bots. So this kinematic replication is not seen anywhere else that we know of in the animal kingdom. It's a completely novel behavior that they figured out in this new context. Again, just skin. And the amazing thing is that they have completely wild type genetics. So this normal frog genome gives rise to embryos that go through a particular stage and they have particular behaviors, and all of these things when you ask why animals have particular form and function, the answer usually is, well, eons of selection, right? So selection has pushed this genome into a state where it produces very specific kinds of things fit to their environment. Well, that same genome can make xenobots. There's never been any xenobots around to be selected for. There's never been selection to be a good xenobot. They have this really weird developmental sequence. They have these strange behaviors. We don't know what their cognitive capacities are. We're investigating that now, whether we can train them and so on. But it really tells you that in the absence of the instructive interactions with the rest of the body, what your skin cells by default want to be doing is this. They would like to be working together to form this kind of proto-organism. And the actual large-scale morphology operates because in the normal context there are signals that force them to have this very different two-dimensional quiescent lifestyle. So this idea of agendas throughout the biological multi-scale architecture is, I think, what's going to drive a fundamental solution to cancer. This is the kind of plasticity that we need to get under control if we really want to solve cancer. So that's it. I will thank the postdocs and students and collaborators that have done all the work that I showed you today. I thank the model systems. They do all the heavy lifting. Our funders, and again, the disclosure for the limb regeneration work. And that's it. I thank you, and I will take questions.
Thanks, Mike. I'd just like to comment, what a wonderful talk. You've got a real knack of communicating some complex science, so thank you for a wonderful talk. There's a couple of questions in the chat to start with, so I'll come to you first, Alvaro. Do you want to come on in?
Yes, thank you, Frankie. Hi, Michael. Thank you so much. What an inspiring and stimulating talk.
Thank you so much.
Yeah, at the beginning, when you were talking about our capacity to regenerate compared to these organisms, I mean, I would argue that I'm not a biologist, I don't work with these organisms, actually. I learned them from you, but we do have an incredible capacity to regenerate in terms of efficiency and reproducibility, right? As long as it is a small scale. So most of our tissues, when there's a fracture or rupture, we're able to regenerate them. So does that mean that the electrical activity of those of our cells at small scales, they tend to behave well, and when you go at higher scales, that's when things get messed up?
Yeah, that's an excellent point, and I think it relates to a question that I get a lot, which is what happens during aging, right? What happens because we all face a decline of regenerative ability with age, right? And our epithelia, I mean, everything just gets worse. It's a good question. Bioelectricity operates on multiple scales, and yes, there's absolutely bioelectrical signals that are involved in wound healing, so maintenance, right? Tissue maintenance. So let's say your intestinal epithelium, your all of those kinds of things. But and as do many other signals, so biophysical signals, biochemical, and so on. I think that there's a real difference. There's a real fundamental difference between wound healing, which means seal gaps in epithelia, basically fill in whatever is already here, right? That's the kind of wound healing, repair what's already here, versus real regeneration, which is the whole organ's gone. There's nothing to fill in because the whole thing is gone, right? And some animals are really great at that, and deer can do it. Deer can regenerate the whole antler structure, but humans, with the exception of the liver, we are mostly doing wound healing, not so much regeneration.
Okay, thank you.
Rumen, do you want to come on in next?
Thank you, Michael, for an incredible talk. It's one of the most insightful talks I've probably ever heard.
Thank you so much.
Cancer is governed by somatic evolution, whereby a cancer cell divides and both cellular copies have indelible marks, whether it be protein coding genetic mutations or epigenetic chemical marks. With reference to your morphogenetic field and software in terms of bioelectricity, is there anything analogous to a unit of inheritance?
Yeah, very, very good point. So I'll say two things. At the risk of pushing back a little bit on that first definition. I mean, I certainly think that this kind of clonal selection model is relevant, and in some cancers it's very relevant. But it's not the only way you get cancer. As I showed you, you can get all of the metastatic melanoma phenotypes with no individual cell. There's nothing wrong with any of the individual cells, really, right? That the hardware is all exactly intact. In terms of the unit of selection, I think it's less about that and more about learning. I mean, one of the things, so people certainly studied clonal selection in cancers and show how it, for example, resists resistance to chemotherapy, right? You can have rare cells that are resistant, and they proliferate kind of like bacterial persisters. That's not the only way. So cells actually learn, right? And this is we could do a whole other talk on basal cognition and cell learning and so on. I think the bioelectrics is more relevant to that. Although I will say there's a piece of this that I had to cut out for time reasons. If one of the things we can do I mentioned that bioelectricity is a kind of memory, and not just because that's where neural memory evolved from, as these ancient kinds of things. But when we can take a flatworm and literally change the electrical memory that tells it how many heads it's supposed to have, okay? And as it turns out, the number of heads is determined by bioelectrical state. The default is 1, but it doesn't have to be 1. And we can go in electrically, we can change it to say that, no, actually correct worms have 2. And from then on, however many times you cut that animal into pieces, you will get 2-headed worms. And that's even though the genetics are untouched, right? There's nothing wrong with the genome. It's because some of this information, and you can think about it as inheritance, because that's exactly how these animals reproduce natively. They just tear themselves in half. So it's a weird kind of inheritance. It's not quite, sperm and egg, but inheritance.
Thank you, Raj.
Then, Rafina, do you want to?
Yes, hi, thanks. And again, it's a really excellent talk. And as someone who's interested in bioelectricity and has been for many years, it was really lovely to be able to showcase how important bioelectricity is. But Mike, I had 2 questions, if you don't mind. One is, we've been doing patching of individual cancer cells, and we can show them to be depolarized, and then we've gone on to do galvanotaxis. I was wondering what you actually think is the long-term cue then. Are you thinking that it is electrical gradients that are modified and then transmitted across cells and tissues? And how is that coordinated? Because we've used a reductionist approach, but everything you've said today suggests that that probably isn't the way forward. So I just wanted your thoughts on that. And if you don't mind, my second question really is, membrane potential is composite of many other ion channel conductances. And again, how would you, I get the point about using ion channel blockers and targeting, but is there a certain amount of redundancy built into how the membrane potential can be controlled? Thanks.
Absolutely. Thank you so much. 2 great questions. So let's do the second one first. The key to all of this, and I think the thing that's slowing down or holding back the use of ion channel drugs as cancer drugs, is that people are thinking of targeting specific channels. They're saying, oh, this channel is upregulated in cancer, I'm going to knock it down the way I would if it was a growth factor or something like that. The thing is, the voltage, exactly as you pointed out, is a composite of lots of different conductances. And in fact, there's lots of redundancy and compensation. The cell will try hard to fight. When you try to change the voltage, the cell will fight. It'll upregulate other channels, open and close other channels. It tries to fight. So the idea is you can never just look at one channel. What you're looking at is what is going to be the dominant, what is going to dominate the VMEM. So which channel can you take advantage of that's going to prevent the cell from counteracting what you're trying to do to it. So for example, the native channel might be a potassium channel, but the answer is there's actually a chloride channel that if you were able to get that open, it would completely dominate everything else the cell was doing, and you can set the cell to a particular voltage. One of the things that we do in basically every paper on bioelectrics is we show that you get to the same outcome as long as you track the voltage, no matter what type of channel you use. So that eye you can use, for example, and the same thing with the cancer, we've shown this, you can do potassium, sodium, chloride, protons, it doesn't matter which channel you use because the outcome isn't tied to the gene. You just have to get the voltage right. And so what you need is a bioelectrical simulator that will tell you, well, which of these things is going to dominate the potential. It gets more complex because it's not just the potential of one cell. So this gets back to your first question. Yes, you can track the single cell properties of these things, but we've shown, for example, that when you induce let's say a KRAS, a nasty KRAS mutation on one side of the animal, I can put an ion channel on the other side of the tadpole, and the inhibition of the tumor rate is exactly the same as if I hit those exact same cells. It does not matter, because what bioelectrical networks are really good at is propagating that information across distance, and we have models that explain how that works. Now, this is all frogs, so the distance is quite small. It's about a centimeter. So you might say, well, in a human, that's still microenvironment, right? A centimeter is not that long. I don't know how it'll shake out in humans, but a centimeter to a tadpole, it's many thousands of cell diameters. So it's really not local at all, and you can't just track the individual cell potential, and I've shown that those melanocytes, I showed that too, right? You can trigger from the other side of the neural tube, and it still works. So I think understanding the way this information spreads through the network is really critical. What are the cells saying to each other to get them to be normal? That I think is the root of all of this.
Thank you very much. Thanks.
Thank you.
I'll ask a quick follow-up question, if that's okay, Mike. So, from a traditional point of view of cancer, which we think of as originating in one cell,
yep,
which then replicates and turns into your tumor. Where does this sit with the context of network communications then? Because it seems counterintuitive that one cell would change the whole network, if that makes sense.
Yeah. Well, that's a good question. I would say two things. I would say, first of all, the whole idea of one cell causing cancer. I'm not going to say that doesn't happen, but I don't think it's the whole story by any means. And I'm not the only one saying that. That story has been cracking for years. There are lots of people arguing against that now. However, having said all that, and we and others have shown that you can trigger it without any cells being wrong. I mean, we had in some of our initial papers, we had a terrible time with the reviewers because they said, well, what's the initiating mutation? And I said, well, there is no initiating mutation. They said, well, then it can't be cancer. I said, well, that's if you define it that way, but that's a very specific definition that may not be right. However, I will say this: we had another paper where we asked the question, when we induce the cancer, what is the smallest number of electrically dysregulated cells that we can introduce, right? So what's the minimum? And it wasn't one, but it wasn't more than a dozen. It was literally a very small, and that's very weird. You would think that why is it that a small number of cells can dysregulate the whole organism? We have some computational models now that can sort of explain part of that would basically, there's a kind of tipping point idea, right? And there's amplification loops and so on. There are key cells that when their voltage goes off, everybody else eventually goes off, right? It's a kind of a tipping point thing, but lots more work remains to be done there. But small numbers of cells can in fact kickstart large waves.
So there's two more questions. Are you okay for time, Mike?
Two more. Yeah, let's do it.
Excellent. Josephine, do you want to come on in?
Hi, Michael. Thanks for your talk. It was really interesting.
Thank you.
I have a question. It's about the ethics. Are you able to foretell any issues around any ethical issues around like bioelectronic medicine in mammalian cells, even though I know you haven't really started on that yet?
Yeah, I mean, we think about ethics quite a bit. I don't know that there are any. I mean, I'm not sure what the ethics issues would be. This is not different from, I think, from other types of medicine. Really, we're talking about. I mean, in the end, we're talking about using the same drugs that people take for epilepsy and for cardiac syndromes and so on. I don't know what the issues might be here. It seems relatively unproblematic to me.
Thank you.
Thank you. Okay.
Last but not least, Donata.
Yes, thank you very much. It's been a pleasure listening to your talk. So I have two questions, very briefly. So you said that you were basically your approach uses electroceuticals, right? And so that you're not instead using, like, for example, external fields. And I was wondering why there, and like, yeah, if you can comment on that. And then if you have investigated, and I guess maybe you have, the epigenetic role, the control at the epigenetic level. Thanks a lot.
Yes, yes. A couple. Okay, so on the epigenetics, a couple of things. So first of all, nowadays, epigenetics is usually referring to like chromatin modifications and things like that, right? The traditional definition of epigenetics is actually all of the information that isn't genetic, in which bioelectricity is actually an epigenetic modality in the traditional Waddington's sense of epigenetics, right? This is a biophysical layer of information that is not genetic. So in a sense, it's already this is epigenetics. However, we certainly have looked for so downstream of bioelectrics are things like HDAC1 activity, so histone deacetylases, right? So acetylation state. All of these things are in fact downstream of just like gene expression and cell movement and so on. With respect to the electric fields, I'll say this: I have nothing against electric field approaches and so on. As far as we know right now, applied fields with electrodes are not a good way to establish spatial patterns of resting potential, right? With electrodes, I can spike electrically excitable cells, right? So neurons, so neuroscientists do this all the time. That's easy. But if I want to establish a voltage pattern where depolarized, hyperpolarized, depolarized, and I want to do that for the next 6 hours, no one has a way of inducing that with applied electric fields, because if you apply an electric field across a cell, what will happen is one side gets depolarized, the other side gets hyperpolarized, the net change is zero. So I don't know with electrodes how to set up a voltage pattern across cells and keep it there, not for milliseconds, which is easy, but for hours. So it's just a technological limitation. I don't think that electrodes are particularly good for that, but maybe someday engineers will, figure something out.
Thank you very much, Vincent.
Thank you.
One final question, Mike, if there's time, just because Sheila, do you want to come on in?
Hello, thank you for a fantastic talk.
Thank you.
It's really made me think. I have a rather simple question. It was just that, have you seen examples of bioelectric communication in the prokaryote world or in the plant kingdom?
Yeah, great question. Okay, so plants, there's a little bit of work on. We don't do it, but there's a little bit of work in plants. There are some amazing stories about bacteria. This is Soel Grohl's work, and he has a number of really interesting. If you want the papers, just drop me an email, and I'll point you to his stuff. Basically, what he found is that evolution discovered the use of bioelectric networks for scaling from single cells to large kinds of goals around the time of bacterial biofilms. So bacteria already do all this kind of stuff in biofilms to synchronize feeding and other things, sort of on their way to multicellularity, but not really yet. Yeah, there's great, and also Adam Cohen had some nice work on voltage in E. coli, I think. So yeah.
Okay, thank you very much. I'll drop you a line. Thank you.
Thank you.
So once again, Mike, just thanks for the time. Thanks for a great talk. I'm sure you've given us all lots to think about, and yeah, no doubt some people will be in touch, Mike, with you. Okay.
Great. Please. Yeah. Thank you so much for the great discussion. Yeah. Please email me if you have any more thoughts.
Brilliant. Thank you. Bye-bye.
Frankie. Bye. Bye, everybody.
Thanks, everyone.